Okay, good morning everyone. Thank you for being here. So it's a great pleasure to introduce Liz-Marzán for the fourth and last lecture of the series. So thank you for being here. So many people are also watching in the internet, the videos. So today Luis will be talking more about applications and bio application, and he will share with us the frontier research that is doing in his lab related to this application. So Luis, please.
Thank you, Marco. Okay, bonjour. Thank you for those who keep coming back, and welcome to the ones who came specifically for this lecture. So unfortunately, yes, this is the end of this lecture series. I'm going to try to guide you through what we feel is a promising avenue toward application of the materials that I have described during the previous lectures. So if this wants to work. Okay, so I already showed you this slide, I think, which is related to one of the most popular integrations of plasmonic nanoparticles into biosensors, originally designed for the pregnancy test and then applied to many other types of sensing based on these lateral flow assays, and most recently used for the so-called antigen COVID tests. So this is a rather simple but very efficient way of doing biosensing with plasmonics, but it's probably not the most sensitive one or the most versatile one. It is very versatile because you can detect many biomarkers, but there are many other things that can simply not be done with this configuration. On the other hand, I also told you about Raman spectroscopy and surface-enhanced Raman, and how by designing either the morphology of the plasmonic nanoparticles or their organization, we were able to induce the enhancement of electric fields next to the nanoparticle surface, so that the Raman signal of molecules attaching to them would be enhanced many orders of magnitude, making this initially per se selective spectroscopy tool also very highly sensitive. And if you remember, so this is an example of analytes adsorbed onto nanoparticles that are isolated from each other. And the red one is what happens when you simply aggregate the same particles, and then the analyte is located at the gaps between the particles. Now, in order to do this, as you have seen, we can make particles of many different morphologies, and we can tune the dimensions, and we can tune the anisotropy, so we can select the morphology that is most efficient for the application, maybe related to the enhancement of the electric field, or adapted to the wavelength that we need to use for our application. And in this respect, you're probably familiar with the concept of optical transparency window or biological transparency windows in the electromagnetic spectrum. And so typically there are three that are defined. You may even define one more here, and which are related to those areas where the biological materials, the biological tissue, absorb or scatter less light, and so you can get better penetration of light and less absorption. For example, avoiding the absorption by water or by hemoglobin in blood, and a few other biomolecules that can hinder the transmission of light in and out of the tissue, which is very important if you really want to apply these materials as sensors within a living tissue or a tissue model. And so, of course, we can do that, and this is just a simple example for nanorods and nanostars, where by simply tuning the dimensions of either the length of the rod or the spikes on the nanostar, you can take the maximum absorption exactly to the area where you have this first transparency window. And more specifically, you can even take it exactly to the wavelength of the laser that we typically use to examine biological samples in Raman spectroscopy, which is 785 nanometers. Another interesting point is that if you want to combine this with photothermal therapy or hyperthermia in general, typically this is also done with laser beams around 800 nanometers, which is within the same window and also coincides quite well with the wavelength that we use for the analysis. So this can all be connected, and thanks to the advances that I showed you in the previous lectures, now we can routinely target the wavelengths that we need. I think I also showed you this slide with the main advantages and still drawbacks that we have for the application of this technique, insisting on selectivity and sensitivity, the use of portable instruments and encoding of nanoparticles, which I already introduced last week, but I will show you some application this week. And also the application to almost every type of analyte, which has to be taken into brackets because depending on the Raman cross-section of the molecule and their affinity to the metal surfaces, you may have big trouble using the technique to identify some molecules that may be of interest. But more problematic than that is being able to make the plasmonic substrates with sufficient reproducibility so that you can measure every time the same type of analyte, the same concentration, and obtain the same signal. This is still very tricky. To make them uniform enough so that if you're analyzing on the same substrate at different spots, you should also get very similar results to each other. Bringing the analyte in contact with the metallic surface, as I said, you may have trouble because some other components of the mixture where your analyte is located may block the surface of the metal, and then you will not have enough enhancement of the electric field for the molecule you want to detect. Or even if your analyte is binding, if there are other molecules around with a sufficiently high Raman cross-section and they also bind to the metal, they may complicate the analysis of your spectra. And also, and this I will also address in more detail today, memory effects, which I repeat, these are related to the absorption of molecules on the substrate at early times that will not leave the nanoparticle surface, and they will contribute to spectra that you measure at later times. And then you will not know whether what you measure is coming from what is happening at that time in the system or from what happened a few hours before. Okay. All right, so let's address reproducibility and uniformity. And then you remember we have access to computational tools that allow us to simulate the optical response of rather complicated systems. And so this is something that I would invite you to read this paper to understand more in detail. It's an implementation of the boundary element method with accelerated computational tools that allow, and also parallel computing, that allow you to simulate in one single simulation run the response of all these hundreds of particles that are located next to each other, reproducing what we have in the electron micrographs. So here our colleagues were really trying to simulate what we typically have on a substrate when we dry a droplet of our nanoparticles, which again, this is, if you use the colloidal methods, this is the most common way of preparing deposited substrates for Raman enhancement. Okay, now what you see here is already the result of the simulation, where the white spots would be those areas where the Raman signal is amplified most, and the dark ones would be the areas where there is no amplification. And so what you can see is that, well, you could say that there are many white spots, but if you calculate the percentage, it's actually very small, okay? And so in fact, when you run a surface enhanced Raman experiment, you obtain the signal mainly from the molecules that are located at those spots. The rest of the surface is almost useless, okay? And in this particular case, you see all these areas where there is sufficient distance between the nanoparticles are basically silent. They do not provide anything to the signal that you record. You can even get in more trouble if you're simulating the response of nanostars. And you remember, nanostars are great particles for enhancement of Raman signals when they are isolated because of the presence of these sharp tips. And then if you're able to bring your molecules to the tips, you will get a really high enhancement. But if you calculate now the response when the particles are getting very close to each other and even entangled, the simulation tells you that you will obtain a sort of destructive interference, meaning that the signal will be lower than what you expect and even lower than what you get for a less dense array of nanostars. And I'll show you that this actually fits with experimental data. So in fact, if you now plot the surge enhancement calculated for spheres, rods, or stars at increasing surface density coverage, you see for spheres and rods, everything makes sense. As you're increasing the density of particles on the substrate, there is a gradual increase in the enhancement because you have more and more particles that are providing this enhanced electric field. And here you have this sharp increase in the intensity because they are almost touching each other, so they are creating hotspots. And as I said, the hotspots contribute a lot more than the isolated particles. But for stars, and here there are two calculations, one for a typical Raman enhancing laser, the other for the position of the maximum that we get for the stars. As you are increasing the density, you either remain roughly constant or even get a decreased intensity at very high coverage, which is in agreement with that destructive interference that I mentioned earlier. And Claire here will remember that we used this same argument to discuss this data in our first collaboration, where we were using nanostars, and then they were measuring the photothermal efficiency of the nanostars of different sizes and at different wavelengths. And they were doing that in water, in cells, and then injecting into mice. And then there were some surprising results that, depending on the time at which you measure, for example, within the animal, the best response would come either from the intermediate or from the larger particles. But this was actually quite counterintuitive because we were observing that at later times, some of the stars were not so efficient, and this was related to their accumulation within the endosomes. So they start to have this destructive interference as well. Okay, now I'm going to show you some fancy substrates because I think they are very nice, but I don't think we will use them very often, okay? So this was a piece of work that we did in collaboration with Rafal Klein. And, actually, most of the work that you will see was done at Rafal's lab, and one of my students was involved. And then we did also the Raman analysis. So this is a rather smart idea to create first binary lattices. So you basically mix two different types of nanoparticles with different sizes, so that when they self-assemble into organized 2D lattices, they will interpenetrate each other. So here you see these are the small gold particles, and these are the larger iron oxide particles. Because of the high monodispersity and the careful choice of the relative dimensions between them, they perfectly organize into binary lattices where both types of nanoparticles are ordered, and they are ordered with each other. And now what they found is that, so these lattices are prepared by a typical method in which you deposit the colloid, the hydrophobic colloid, with the two nanoparticle types on the surface of a higher density solvent, immiscible, like diethylene glycol. And then you let the organic solvent evaporate, and then you form the monolayer on at the liquid air interface, and then you can sort of fish it with the solid substrate and take it away. Now, if they were doing this with a surface that was coated by a carbon film, like the TEM grids that we use typically, then by simply heating it up a little bit and then irradiating, for example, in the electron microscope, the structures were much more robust than at the beginning. Meaning that now you can do chemical etching to remove either one or the other component. And then you can get these very unusual 100 organized lattices that are very difficult to do by standard self-assembly. And so you get them for both components, depending what you use to etch away the nanoparticles. So with hydrochloric acid, you would not dissolve gold. You would only dissolve the iron oxide. With cyanide at the right pH, you would dissolve gold, but not the iron oxide. Okay. Now, the nice thing is that you can do this not only for these monolayers, but actually you can increase the concentration of the particles and obtain multilayers. And then it becomes interesting because depending on what the concentration is, then the ratio between the different types of particles, then you can get a lot of different structures where you get combinations of gold particles and air into crystalline lattices, which Rafael wanted to call nanoallotropes, because these are sometimes organization of gold, which is not even known in nature. Okay, so these are very nice, and our colleague Peter Krall in Michigan, St. Louis, so he actually did some simulations to explain why we could obtain this type of assemblies and how they could get organized into these multi-layer lattices. And then our colleagues in Antwerp did all that electron tomography, and we can see, for example, these movies where you can see that the lattices are not only ordered, but leave a lot of empty spaces. So if you think about surface-enhanced Raman, this would actually be very nice because the analyte molecules could diffuse through the big pores and then deposit at the gaps between the nanoparticles everywhere. And the films are thin enough that you will obtain a signal from every layer in these multi-layers. And so this is what you see for different assemblies with more and more layers. For the monolayer, there is nothing because the particles are too small and too far from each other. For this sort of, I think it is a tri-layer, you get already a decent surface-enhanced Raman signal. And then if you increase the number of layers, then you get much better sensitivity. Now, this unfortunately can only be done in very small sizes, and it's, I would say, a bit complicated to obtain properly. So there are other ways of making well-organized assemblies of nanoparticles. And I'm going to show you quickly one that we already discarded, but which was very useful for some time, which is related also to the theoretical predictions of our colleagues. And so we asked them to compare the efficiency of thick multi-layers of gold nanorods, which are deposited with random orientation and on top of each other, and roughly the same number of rods organized into multi-layers that are standing perpendicular to the substrate. Well, you can imagine that this was not a random choice, and we had already a hint that we would be able to prepare that type of substrate. But anyway, the major result is not only that there are differences in the efficiency at different wavelengths, it's mainly that here, this is the calculation for the efficiency at very close to the top of the substrate. I don't have here, but we also have the simulation for a few tens of nanometers inside of the film. And here, almost everything becomes black, meaning that here you have, again, a lot of destructive interferences, and therefore the efficiency of the interaction of light with the inner layers is much lower than what you would expect. However, for the multi-layer system, at every plane in between monolayers, you find again a simulation like this with plenty of hotspots. So now the challenge was, can we prepare, first of all, ordered arrays of gold nanorods like this? And second, will we be able to bring our analyte in between the monolayers? And so this was a big part of the first ESC grant that I had related to understanding the communication between bacteria. And so we decided to use this sort of combined top-down and bottom-up approach, where by lithography we could obtain these PDMS templates with rather big holes, I would say, micron size, and then basically use them as a template to guide the organization of nanorods inside of each of these cavities, and then let the solvent gradually evaporate and obtain the multi-layers upon removal of the PDMS template. And then after doing that, you obtain a dark field optical microscopy image like this, which at least shows you that the organization of the nanoparticles has somehow followed the morphology of the holes that we had in the PDMS stamp. And now you come with these samples into the scanning electron microscope, and then you find pictures like this, where actually, so this is already with a very well-defined substrate template, where all the holes were the same size and very well organized in the space. And so here you see there's these towers that you see very well uniformly spaced and very well defined are actually made of multi-layers of nanorods. So this movie is made by coming with a focused ion beam, cutting planes very finely and taking SEM pictures at every cut. And then you can reconstruct three-dimensionally, and you find that we get something which is very similar to what was calculated, and that was giving us this reproduction of hotspots at every interlayer. Now, managing to get a good signal here was not straightforward, and we actually had to combine these substrates with a salt gel process related to creating a matrix that would protect the towers. But we were so lucky that the silica, the mesoporous silica, was actually infiltrating the multilayers and creating little gaps between the monolayers with mesopores through which the analytes could diffuse. And not only that, the small metabolites that the bacteria used to communicate with each other were able to diffuse through the pores, but the pores were small enough that proteins, lipids, and other bigger molecules could not diffuse inside. Okay? So this is basically the idea that we had, where we identified bacteria that have this communication cycle with some analytes that have a rather high Raman cross-section, and then we could do quite a bit of analysis even of intercommunication between different bacteria families. I don't have time to explain this today. I would like to go more into the more recent research that we are doing. Okay, now Cyril Amon, whom I expected to see here today, but apparently he couldn't. So he was here the first day. He was a postdoc in my lab at that time. He's now Maître de Conférence, I think, at Paris-Saclay. And he had experience in making these multi-layers, and he did a good job already in the initial part of the work. But then, because also of the simulations, we started to think whether we need so many nanorod layers and whether this is the most efficient way to do the analysis. And so again, we did some theoretical work with our colleagues, comparing monolayer, bilayer, trilayer, tetralayer, and then calculating the spectra and also the enhancement of the electric field for these multi-layer systems. And what we find is that in terms of efficiency, there is not really a big advantage in increasing gradually and even more the number of layers. And in fact, Cyril was able to make these samples experimentally. So this is, again, electron tomography showing one of these bilayers on a substrate. And this is actually a picture from the top of the bilayers. So you see they are very well organized in this case when there are very few layers. And so in fact, it was lucky that during one of these depositions, there were these parts of the substrate where you could identify monolayer, bilayer, trilayer on the same region, and which was big enough that then you could come with the laser beam and focus on the different spots on the different layers and then obtain the Raman signal for the same molecule. And what we found was that for the wavelength that we are typically using, there is almost exactly the same intensity for the three different systems. And so then we started to think, will we be able to obtain templates that will allow us to combine now what we know that probably less layers can be more efficient with templating and getting a uniform distribution of these multi-layer systems on a big substrate. And this is when we were lucky to meet Agustín Mee, who is now he's a research professor at the Spanish Research Council. And so with him and his theoretical colleagues, we decided that an array of clusters like this would be very efficient. And then, well, so of course we calculated because we knew there was a good chance we could obtain that. And the idea was this. So exactly the same thing as I showed you earlier, but now Agustín can actually prepare these templates with holes that are only a few hundred nanometers in diameter, a few tens of nanometers in depth, and a few hundred of nanometers in separation. And this is very interesting for several reasons. One is we can obtain these very well-defined arrays, of course, after optimization of the conditions. Second, we can do it with spheres, but also with nanorods. I am not showing this today because it would become too long. Third, because when you're organizing the particles like this, so each of the dots that you see here would correspond to something like this, where you have crystalline order within each of the spots that then create crystalline, supracrystalline order in 2D. And now, because of the dimensions of the lattice parameter here, we have not only a plasmonic response, we also have optical diffraction. So this is effectively a 2D photonic crystal, where now you can create, so you see these bright colors are typical for opals and related systems, meaning that there is light diffraction. And in fact, if you tilt the sample, the color will be different because the diffraction condition will be different. But the interesting point is that the simulations also tell us that when we do this, you create so-called lattice plasmons, which actually propagate on the surface, and they contribute to a further enhancement of the electric field at the gaps between the particles in each of the spots. Okay? So this is a system that we like very much and that we thought could be the starting point for the project that I would like to describe today. So the project looks like this. It's called 4D BioSearch, and it proposes a very challenging objective, I think, and the referees also thought so, which is, let's try to use surface-enhanced Raman to understand the behavior of tumors. And we propose to do it by combining two strategies, which are both related to surface-enhanced Raman. One is building biocompatible scaffolds by 3D printing, which would be made of hydrogels that will be affine to the growth of the cancer cells and other components of the tumors, and will carry inside plasmonic nanoparticles so that you would be able to do sensing of biomarkers related to the activity of the tumors. And in particular, we were very much interested in analyzing the deregularized metabolism of cancer cells, because this leads to changes in metabolites, and these metabolites are small molecules that we like to measure with surface-enhanced Raman. And they are actually very important in understanding the behavior of the tumors. Second, we would prepare many labels, or as many labels as needed, I showed you how to do that last week, by using small particles labeled with different Raman active molecules, which would then be used to label cells that would come up within these 3D models. And then we should be able to watch over time the behavior and the distribution of the cells by distinguishing, for example, cancer cells from fibroblasts and a few other, and even if time allows and we are able to do that, to watch the differentiation of cancer stem cells. Okay, so I'm gonna tell you a little bit about the progress that we have made. Actually, when I wrote the abstract for this lecture, we had almost not even started this work because it was pre-COVID. Now we have a few results that I can show to you. Okay, so let's start by seeing if we can monitor metabolic processes in cancer cells. And so we started by a rather simple process, which happens actually in many types of cancer cells. In this case, we used the most standard HeLa cells. And this is the catabolic conversion of tryptophan into kynurenine. So these are two metabolites that are transformed in cancer tissue. And so the idea is that now we can take samples from the supernatant of the cancer cells, and then with our substrates we should be able to identify both metabolites. This will come later, and to watch how one converts into the other over time. So first of all, we measure the Raman signal and the surface enhanced Raman signal of the pure molecules. We find a big area where it's a bit more difficult to distinguish, but we have clear peaks that would serve to distinguish one metabolite from the other. And we actually also show that this can be recorded properly within cell culture medium. And then, well, there are many more results that I could show you, but this is one example where by taking samples at different times and measure on top every time of a new substrate, we see how the peak of tryptophan is decreasing in intensity while the peak of kynurenine is increasing in intensity. Another piece of work that was related to this type of studies was actually watching what happens when you induce cell death. You would expect that there is release of quite a number of molecules, including purine derivatives. And actually, to do that, Javier Plou, who is a student who finalized a few weeks ago and is now staying for one more year to continue this work, he designed this simple cancer on a chip model where, again, the cancer cells could be cultured on top of, in the bottom of this very tiny chip. And then we could place directly our 2D supercrystal substrate on top with the particles facing the supernatant. And then we would be able to do the analysis directly in situ. And so by doing this, he was observing not only the conversion of the tryptophan to kynurenine, but also this other peak that initially we did not understand. This is why it was labeled like U unknown. And then it was identified as coming from these purine derivatives, and it was shown that by inducing cell death at different concentrations of hemine, we could actually observe the increase of the intensity of this peak, and at this rather high the concentration, we would also be able to watch the increase of this analyte over time. So this opens up perspectives, and we are now pursuing other a bit more complicated processes and doing all the controls with the standard techniques that our colleagues at CIC bioGUNE, experts in cancer metabolism, are typically using to analyze these samples. But what we are finding, and I hope this will be wrapped up very soon, is that with this faster and more simple way of analyzing the behavior of these cancer cells, we are starting to observe metabolic processes that are not so well understood so far in other systems. Anyway, one of the problems that we had was that in order to do this, as I mentioned, we were using one substrate for each measurement, meaning that if you're using this cancer on a chip model, you would have to do something like this. So you would have to first come and very carefully lift the substrate to remove it, and then come with one more, very carefully, you see shaky hands anyway, and place it again on top, trying not to disturb the cell culture already in the well, and trying not to take liquid away when you're removing this one. So not very comfortable. And so again, Javier, together with another student, Matthias Charconnet, they came up with this idea, or trying to solve this problem by implementing a very practical solution that, in fact, we filed a patent to try to protect this technology. So the idea is, if you have simply your gold nanoparticle superlattice, and you're incubating it with one analyte like mercaptobenzoic acid, mercaptobenzoic acid will quickly bind to the gold particles through the thiol group, and will show this very well-defined peak that is characteristic of the vibrations. Now, even if you clean the substrate and then you dip it again in a solution of another molecule like nicotinamide, which would also like to bind to the particles, and then you measure, you still only see the signal from the NBA molecules that were initially attached on the nanoparticles. And the same if you clean it and incubate it with a mixture of the two analytes. So now the idea is we have to manage to obtain something like this, where we can cover the nanoparticles by a protecting layer, and then open a window, do the first measurement, and then clean, open another window and measure the second analyte and obtain this peak, and then clean and measure on another window and obtain the two peaks of the mixture. Okay? So this can be done by using thermosensitive polymers, meaning thermally degradable, like PLGA, polylactic glycolic acid. Okay? So you can prepare PLGA with different compositions so that it's more or less robust, more or less degradable. But in most cases, if you can use it, for example, to do this spin coating and deposit a micron thick layer of the polymer on top of the nanoparticles. So this is an SEM image of good quality, but now the polymer is covering the nanoparticles. Okay? And now, because you have the plasmonic nanoparticles below, you can come with a laser beam, focus it on a tiny spot, and then degrade locally the PLGA so that you're effectively opening a window where the particles here are now available to anything that is coming from the solution. And the rest of the substrate is still protected by the polymer layer. So you will not get any signal from other areas on the substrate. And so if you do that now, the way I told you, so you first incubate with one analyte, open a window, then clean, incubate with another analyte, open another window, clean, incubate with third analyte, open another window, and then you can even come at the end and do your Raman mapping, and you will obtain the three different spectra at the three different spots, each of them corresponding to the analyte that has bound to the particles at its specific time. Okay? So this is actually quite nice because now you can really think about real-time measurements without caring about replacing the substrate every time. And so, for example, you can implement it in a real microfluidic chip where you can flow your analytes at different times, and then every time you want to measure a spectrum, you basically either move the substrate or move the beam, and then irradiate at higher intensity, and then measure at a lower intensity of the laser. And so you can do that for many cycles. And so these are examples with two different analytes and flowing with water, so that you measure one or nothing, one or nothing, one or nothing, and then the other or nothing, and so on and so forth. Okay? So you can every time measure a new spectrum corresponding to the solution that you're flowing through your system on top of your substrate. And then coming back to the example that we had, this is again a cell culture where now we are observing a different metabolic conversion from adenosine to hypoxanthine. And again, if you do it simply with the superlattice substrate without the polymer film and without replacing it, basically you measure the same spectrum at initial time and after 1 day. If you take the supernatant at the 2 different types and use 2 different substrates, you see the conversion. With the PLGA, same substrate, open 2 different windows, you see the same conversion. And now you can do this. So actually we do this now quite routinely to do measurements over time and monitor processes in different aspects. Okay, let's move to the next step in the project. So now we know that we can monitor, at least in the ideal conditions, we can monitor metabolic processes. Now we want to do it in a system that will reproduce as well as possible what happens in a human being. And so, as you know, it's this initial cell culture studies are done basically depositing cells in 2D on a cover slip. Or you can create so-called spheroids, so the cells aggregate with each other and form like bigger entities, which are, well, at least giving some idea of what is happening in 3D in the real systems. But you can also create more complicated structures, as I indicated initially, by building scaffolds and then incorporating cells, different types of cells, and also other components of the stroma, collagen, and a few other things that you have typically in these tumors. And this would be something in between what happens in the 2D and what happens in the patient or even in animals. And so, trying to fabricate something that is intermediate between these 2 extremes, we came up with using 3D printing, which of course is not a very new idea. Many people are doing that, and there are plenty of people who are doing much better than us these organoids. But we wanted to do it adapted to the type of study that we want to carry out. And so we started by buying a bioprinter and then by formulating inks that, in our opinion, would lead to well-defined hydrogels that could be reinforced by post-printing treatments so that they would be mechanically stable, and then we could manipulate to do either analysis or growth of cells or both combined. And so this is one of the examples where we have this hyaluronic acid together with kappa-carrageenan, and then we include in the formulation gold nanorods. Before all of this works, you have to do all the rheology analysis so that you can understand the flow properties of the system, so that they will do the expected shear thinning, so that when you apply pressure, they will become more liquid, flow through the tip, and then upon coming out of the flow, they will rigidify again. Anyway, another important point is that the nanorods at the end have to be available to the molecules that come from the solution around, so that they will bind to the rods and we can do the sensing. And so these hydrogels that we are printing have to be porous enough so that at least the small molecules can diffuse through. And this again leads to a new series of optimization there. Okay, anyway, so after quite a bit of work of my students and postdocs, we managed to obtain this type of multi-layer scaffolds. The resolution here is of micron scale, also because we want cells to be incorporated here, so you need enough space for them to grow and even to divide and so on. This picture here is one example of ultramicrotoming piece of the hydrogel, where you can see that the nanorods are well distributed and in principle available for the molecules to come. And now the challenge is, can we incubate with one of the interesting molecules like adenosine, a typical metabolite, and obtain the Raman signal. So if you do it including water, this is actually quite nice. You obtain a very well-defined peak at the position that you expect. If you do it in the cell culture medium, well, it's not so well defined anymore. It's closer to the noise, but still observable. And depending on the concentration of the nanorods that we use here, we can also improve the quality of the signal. Now, the nice thing that we obtained, and which was not so obvious, was that we can now do confocal Raman. And then we were able, by using this signal as providing the pixels in the analysis, we were able to reconstruct the three-dimensional morphology of the scaffold from the Raman signal. And remember, the signal is only coming where the molecules that we're measuring have come into contact with the nanorods inside. So this image here is actually very revealing. It's showing not only that we can obtain the signal, it's showing that there is a very uniform distribution of the nanorods everywhere, so we can do space-resolved measurements within the same scaffold. Okay? Now, the second part of this 3D work, the Raman reporters. I already told you about this last week. So you have plasmonic nanoparticles, Raman active molecules that will bind to the surface and provide the code that you need for identification. And then you cover it with a protecting layer, and then you can, if you need, you can also include antibodies or other biorecognition moieties. Last week I showed you examples how to make this type of labels with rather big particles. Nowadays we typically use this strategy, which is sort of adapted from my initial work in the group of Wolfgang Parak. Basically, our trick is using now hydrophobic molecules which have a rather high Raman cross-section, like with aromatic groups, carrying also a thiol that will bind to the nanoparticles. So these nanoparticles will become hydrophobic. And then by addition of a block copolymer, which is generically called PMA, then this will interdigitate and wrap these hydrophobic particles, leaving now hydrophilic head groups on the outside, so they can be redispersed in water and in biological media. And so, yeah, this is what you see here. By doing negative staining in electron microscopy, you see this white halo around the nanostars, showing where the organic molecules are surrounding. And this is one typical example of the spectrum of nanostars coated with, in this case, 2-naphthalene thiol and incubated inside of cells. So the red color here comes from areas where this spectrum is localized. Okay? Now, as I mentioned last week, the nice thing about this system is that now you can select different molecules, and then you obtain a library of codes. And these are very well defined. So far, we have been working with analyzing specific peaks for each of them, but now we are also implementing more sophisticated data analysis so that we can use the full spectrum for identification of the signal and differentiation of the different tags. By doing this, also you can internalize now these particles inside of the cells, and they, as I indicated, they usually go into endosomes. You can do it with nanostars. You can do the same, exactly the same synthesis with nanorods or other morphologies. And then you can co-culture different cell types that have been labeled with different encoded particles, and you can monitor how they evolve over time. So this work was done on a 2D system, which was like the preliminary study to then go into 3D. And 3D in Raman is quite tricky because the instruments only recently are really prepared for that, and also because you need a very good sensitivity. And this is provided only, or not only, but it's provided in this case by the efficiency of the enhancing plasmonic particles. So, yes, we have done already confocal Raman. This is an example where we have, this is the substrate, and we have here 2 layers of cells which were labeled with 2 different Raman codes. And over time, you see that what was initially 2 well-separated layers has become like one invading the other. So now we can do this type of studies over time where we observe hopefully even invasion of healthy tissue with cancer cells. So this is really going on right now. We are starting to get the first results about the design of 3D tumors where we can get distribution of cancer cells surrounded by epithelial tissue so that we can start to observe this type of phenomena. The way to do this is, as I said, by now incubating in the same scaffold the analytes and the cells. And so here, this is something, what you see here is what you would expect if this should work. Meaning that now with the same laser beam, you're exciting at the same time the Raman signal of the analyte, which goes inside of the scaffold, and the Raman signal of the Raman tag that is labeling the cells. And so here you see in green, this is how we label the signal of the adenosine coming into the scaffold. And in red is the label that we included in the cells that are now binding around the walls of this scaffold. So this can be done like this, trying to promote the binding of the cells on the scaffold, or you can use an environment for the cells like the commercially available Matrigel, so that you can have more isolated cells inside of the holes. And this is something I would like to show you to wrap up this part of the lecture. This is a piece of work that we did to try to show the potential of these systems to evaluate the efficacy of anti-cancer drugs. And so again, one of these 3D printed systems, the composition here is a bit different. In fact, the incorporation of alginate has turned out to be quite important because it provides a much higher negative surface charge to the scaffold. And then we see that in particular the analyte that I'm going to show you in a minute would diffuse more effectively and be able to target the nanoparticles inside. But anyway, the strategy is exactly the same. So ideally we have the nanorods in between the polymers within the hydrogel. And then again, by building this scaffold within one of these microfluidic chips, the idea is that we plant the cells at every of the holes within the scaffold, surrounded by the Matrigel that will keep them happy if nothing else happens. And then we have here a little reservoir on which we should be able to inject the drug that we want to screen. And so if we are able to correlate, first to identify how is the diffusion of the drug through this system, and second, how that correlates with cell death, then we are on the initial position to analyze the effect of drugs on these 3D cell cultures. And so for this first proof of concept, we decided to work with methylene blue, mainly because it's an efficient Raman scatterer, but also because it's really being used as a photoactivatable anti-cancer drug. So this drug is not toxic or almost not toxic under normal conditions, but if you irradiate at around 660 nanometers, then it becomes toxic because it generates radical oxygen species. Okay, so again, this is a new confocal Raman image showing that methylene blue also internalizes quite well within the scaffold and provides a sufficiently strong signal that we can use to reconstruct the 3D structure of the scaffold. Now, again, learning how to do this, but in the end it worked, that we can incorporate a mixture of Matrigel and cells within the holes of the scaffold. So here we prepare the gels so that they would express the green fluorescent protein, and then we can easily identify a rather high density of cells within the holes of the scaffold. So every green spot that you see here corresponds to either a single cell or an aggregate of cells. And this shows that we have a distribution that then we can monitor either with confocal Raman or with confocal fluorescence microscopy. And then these are some of the initial studies, basically to see by Raman the diffusion of the drug through the system. So this is the sketch where I show you this would be the reservoir where we inject the methylene blue. This number 1 is this area that we watch here, very close to the reservoir. So at different times you see how the intensity is increasing when you try to target the methylene blue. Only exactly at the places where you see the scaffold. Okay, so outside of the scaffold you are not measuring anything because there are no particles that would enhance the signal. In area 2, at this concentration of methylene blue, the diffusion, it takes longer for the signal to be recorded because the diffusion takes a while. So actually this serves us also to identify which would be the appropriate concentration of methylene blue so that you can monitor the process within a reasonable time. And so after adjusting this concentration, we do this control, meaning that we have only water wetting the scaffold, and then we inject the methylene blue, we let it flow for some time, and then we irradiate. And so after 1 hour, we already see signal of the molecule everywhere in the scaffold. So in this case, the diffusion is quite fast. The intensity is increasing over time, basically because you have more and more molecules diffusing now from the solution inside of the scaffold. So there will be more surface enhanced Raman scattering events at every spot, and then we get a higher intensity. When we have the matrix gel embedding the holes, then we see that the same thing happens, but much more slowly. So initially, after 1 hour, you only see signal very close to the reservoir, and then it takes a few hours to really diffuse everywhere and provide the signal everywhere. And this is related, of course, to the viscosity of the matrix gel, which slows down the diffusion of the molecules through the system. And now, the presence of matrix gel and cells brings up a new component, because now you see the diffusion is not only slow, but actually the intensity that you get at the end is also much lower. And so what we reasoned was, here we have not only a higher viscosity, but actually a big part of the methylene blue is internalizing inside of the cells. And this is why it's not able to get to the particles and provide the signal. And so this is quite nice because now we can watch how the cells will die if we do the irradiation. And so this is what you see here summarized. So this is this is the intensity of the Raman signal over space after 2 hours for different concentrations of the methylene blue or the control. So you see, it's going slowly. For a higher concentration, after 24 hours, we see the signal everywhere. Now, regarding cell death, what we did was use the standard dyes to monitor live or dead cells. And then you see for the control, after 24 hours, you see a few cells have died, the same more or less everywhere. But after injection of 20 micromolar methylene blue and irradiation with the red laser, then we actually see that there is a much more cell death, especially at areas closer to the place of injection. So now we can do this correlation, and you see this, this change in the proportion of dead cells over space for a particular time of irradiation. So I think this is a good start point to now start thinking about a bit more complex structures and also more a wider variety of anti-cancer drug that can be tested. Okay, so if you are still strong enough to listen for 5-10 more minutes, then I will tell you a completely different story, which is, oh sorry, this was the conclusion, that we are trying to target this transition between the current universal medicine into future personalized medicine by incorporating surface enhanced Raman to the techniques that are used to screen patients and classify for specific treatments. Okay, so now I go to the last part, which is again related to plasmonic chirality that I also introduced in previous lectures. Okay, so rather than using single particles with chiral structure, this project is related to organizing particles that are non-chiral individually into structures that are chiral. For example, people have used mainly DNA origami, which can be functionalized, and then you can bind particles to specific spots and create, for example, helices of spheres, or these rotating ladders of nanorods. In all cases, the chirality of the superparticle structures give rise to circular dichroism at the wavelengths that are specific for the plasmonic particles. So we did some work related to this some time ago, related to organizing gold nanoparticles onto, say, more mundane templates, and this was simply a gel that was created by the incubation of this molecule at the right concentration. So this was provided by colleagues in Zagreb, and we obtained both enantiomers, so we could create the gels which were rotating either to the right or to the left. And these we wanted to use as templates to organize gold nanoparticles into helices. And so we started with nanorods, and we found structures like this, which did not look very helical to us. But when comparing the optical signal, the optical activity of these samples to either a free solution of nanorods or even a similar structure with spheres, we actually were surprised to see that these red and blue curves correspond to the mixture of the nanorods with the gel. So very high intensity, and these peaks here correspond to the actual signal from the gel itself, but these two other correspond to the plasmonic chirality. And you don't see the same thing for spheres. These are the dotted lines. Here you see only the chirality of the fibers, but you don't see the chirality of the plasmonic system. And so this was quite intriguing, and so we decided to go to the theory, and I was lucky to have a postdoc there at that time, Baptiste Augier, who is now a lecturer in New Zealand. And he actually took the model that was developed by Alexander Govorov to basically adapt the coupled dipole model to these plasmonic systems, which shows very clearly that for spheres, if you have 2 or 3 spheres, even if you try to follow a helix, they are not chiral. They basically fall into a plane or a line. So you need 4 spheres to provide a circular dichroism signal. But for elongated particles like ellipsoids, even with 2 ellipsoids that are at different plates and tilted with respect to each other, you already have chirality. And so with 2 particles, you already obtain the CD signal. So this was our explanation to the signal that we saw there. Even if there is no long-term helical structure in the system, you have many pairs of nanorods that are following the same sort of orientation, so the same handedness in terms of pairs, and so the collective signal coming from those couples adds up to the signal that we record from the collective system. Now, can this be used in practice? Well, in this implementation, maybe, but it's not very clear. And so we started a project a few years ago by the hand of a talented postdoc who obtained a Marie Curie with this Marie Curie fellowship, with this project where we proposed to use this sort of plasmonic chirality to detect the formation of the fibers that are involved in many neurodegenerative disorders. So these are so-called amyloid fibers, and it's quite well accepted that the origin of these diseases is related to some degradation of these proteins, and then aggregation into small aggregates, oligomers, then so-called protofibrils, and then very long and rigid fibers, which would accumulate mainly at the substantia nigra in the brain and promote these diseases. Of course, there is still debate whether it is the long fibers or the protofibrils or even the oligomers which are the most toxic species. And therefore, it's actually important to have techniques that allow you to actually monitor these processes from the very early stages. Maybe not to extract the substantia nigra from the patient and analyze, but maybe to do some analysis either from other fluids or to do research on this topic. So anyway, the idea is that because it is well known that some of these fibers display a double helical structure, and the dimensions could fit with the smallest nanorods that we can prepare, then we would hope that there would be this binding and we could monitor the presence of the fibers by measuring the circular dichroism of the solution. Okay, so we started by using this model protein, alpha-synuclein, which is not only well defined, but actually is involved in some of the most damaging diseases and can be expressed by incubation of bacteria. So it can be basically using the standard protein expression methods, you can obtain well-defined alpha-synuclein, and then by treating it by literature-described methods, you can promote the aggregation into very well-defined fibers. And now you can take these fibers at different incubation stages and mix them with the gold nanorods, and then you can start to see this circular dichroism signal. So the way we do this is, as I mentioned, we take the protein, we incubate it in the conditions that it should scramble and promote the formation of the fibers, and then we take samples at different times, we mix them with gold nanorods, and we measure the CD after equilibration. And we see that at different times the intensity of the CD is increasing, and then if we take samples at different times and place them in the electron microscope, we also see the formation of longer and longer fibers that carry the gold nanorods into a linear fashion. And now the question is whether these are actually double helices as we expect. And if you don't have any other resources and you are very optimistic, then you go to places like this and you find that apparently the nanorods cross each other. I don't know if you agree, but I looked at these pictures many times and other pictures. But to be convinced, we actually sent the samples to our colleagues to do electron tomography. Initially, they didn't really see anything meaningful because the samples were collapsing because of the soft nature of the protein aggregates. But then they did cryo-electron tomography, and then they were able to provide us with several movies like this that I think are convincing enough. They were convincing enough for the referees of the PNAS. And I think, so we are currently working on similar systems to try to expand the library of proteins that we can apply. But we also did some ex vivo studies. So we teamed up with the group of Joaquin Castilla at CSA bioGUNE, who is an expert on these neurodegenerative disorders. And he accessed the local biobank and requested 4 brains from people who died, 2 from different diseases, 2 from Parkinson's. And then they extracted the substantia nigra. Don't ask me how. They homogenized the samples, purified, and then Jatish came with the nanorods, mixed them, and then measured both the linear and the optical activity. And he found really amazing that the control brains did not lead to aggregation or to any meaningful CD, but the 2 Parkinson's brains brought this broadening and very well-defined CD signal. Unfortunately, we were not allowed to bring these samples to the electron microscope because these are infected samples, so it's high biosecurity. We have similar examples for samples with prions, which are also very damaging, and we also see CD. We have lots of work ahead to really demonstrate that this can be applied in general, and hopefully that we can also do this in a more selective manner than we have done it in this example. So with this, I conclude. Thank you again for coming back, and well, maybe on another occasion we have more time to discuss, but for now, I conclude here. I'll be happy to answer questions.